So now we shall talk about elongation of protein synthesis. Like initiation, elongation also requires three protein factors which we call as elongation factors. That is EF, which means elongation factor. The first elongation factor is EFTU, which directs the next tRNA to its correct position in the ribosome. The second elongation factor is EFTS, which is important for regeneration of first elongation factor, that is EFTU. The third elongation factor is EFG, which is also known as translocase, which mediates translocation. So I can discuss about the functions of these elongation factors while discussing different steps in elongation. So let's have a look here about our first elongation factor, that is EFTU. So EFTU, it binds to GTP. This EFTU GTP, it binds to an amino acyl tRNA, which means a tRNA which is already bound with an amino acid. So this EFTU will direct the correct binding of the second tRNA. We are now talking about the second tRNA because in the initiation we have seen the first charged tRNA has entered the P site of the ribosome. Now this second tRNA, which is directed by EFTU, should reach the A site of the ribosome. It should reach the A site of the ribosome because P site is already it is filled by the initiator tRNA. Right. So this EFTU, this elongation factor, once it reaches the once it reaches the P site, I'm very sorry, once it reaches the A site, the GTP is cleaved and GDP is formed, and this EFTU is released from the tRNA. Right. So this EFTU, I said GTP is cleaved. Now it should, to start again the process, I mean to help another tRNA to reach its right site on the ribosome, EFTU should bind to one more GTP. Only then it becomes EFTU GTP complex. So this regeneration of EFTU is mediated by the second elongation factor, that is EFTS. Okay. Now we shall talk about the steps in elongation. See here, you can see that in the P site of the ribosome, you have the initiator tRNA. So this is the A site which is vacant now, means the second tRNA should enter this site. So I told the second tRNA entry into the A site is mediated by our EFTU elongation factor TU. This helps the amino acyl tRNA to reach the A site. Once it reaches the A site, a bond is formed between your first amino acid and the second amino acid. The first amino acid, it gets detached from its tRNA and a bond is formed between your first amino acid which was there in the P site and the second amino acid which was there in the A site. So this bond formation is mediated by an enzyme called as peptidyl transferase enzyme. This peptidyl transferase enzyme, it's not a separate enzyme. This enzymatic activity is present within the larger subunit of your ribosome. Okay, so you can see the peptide bond is formed between the amino acid present in the peptidyl tRNA site and the amino acyl tRNA site. Right. Now what exactly happens is the ribosome will move exactly three nucleotides ahead, three nucleotides ahead on your mRNA. This process is referred as translocation. So please observe here, you can see AUG which was there in the P site, you can see ACA which was there in the A site. Now this ribosome, it literally moves this way, so that AUG now falls in the E site. Here you can see in the E site you will have AUG. Now ACA, which was previously there in the A site, now comes into P site. GUG, which was previously not there in any site of the ribosome, now falls in the A site. This movement of ribosome, three nucleotides ahead on the mRNA, is referred as translocation, and this process of translocation is mediated by our other elongation factor, that is EFG. Now again what happens, one more tRNA which is complementary to GGU should come to this A site, okay, and it should make a bond with this the chain of amino acids. Then again translocation would happen. So in this way the protein chain elongation will continue. The last step is termination. Termination generally occurs when the stop codons are found on the mRNA. The stop codons include UAG, UAA, and UGA. This UAA is these are also known as amber codon or ochre codon, and sometimes also called as opal codon. UAG is called as amber, UAA is called as ochre, and UGA is called as opal codon. When any of these three codons, any one of these three codons are found on the mRNA, the process of translation would stop. Let us discuss how exactly these three codons help in the termination of the translation process. So, like we have discussed in initiation and elongation, here also we have three important protein factors that aids termination, that is RF1, RF2 and RF3. RF1, it functions in recognizing the termination codon, that is UAA or UAG. RF2, it functions by recognizing UAA or UGA stop codons. RF3, it stimulates the dissociation of RF1 and RF2 from the ribosome after the termination. So in this picture you can see that UAG, a stop codon, was encountered. When such stop codon is encountered, the release factor, I mean RF factor, can recognize this stop codon, and now instead of the tRNA, these release factors would come to the A site. They will enter the A site of ribosome and they attach here, because of which the protein chain, the synthesized protein chain, will cut off and it gets released. And now these two ribosomes will get dissociated because of one more important factor or a protein which is called as ribosome recycling factor.